Excellent. Well, thank you, everybody. Thank you, Anisha, and to the NASH Summit for inviting me today to talk about NIMBLE. I think we've heard some very interesting talks today, and I'd like to kind of bring it together as the last talk here to just talk about how we've really to approach non-invasive biomarker development and qualification in NASH. Team science seems to be the key. We are getting a lot of interesting information from various sources in this very enriched field. And today I'd like to focus my talk on how we build these types of partnerships and how they are delivering results. And so I'm the foundation for the NIH. I direct metabolic disorders programs, includes NASH and other metabolic diseases. So I'm delighted to come here today and talk to you about one of our flagship qualification programs in NASH. So I thought today for the agenda, I'll cover three different elements. The first is just a reflection on why NASH research and biomarker development is a fit for public-private partnerships, large consortia model. I'll then talk to you a little bit about the foundation for the NIH and the Biomarkers Consortium and how we bring these types of partnerships together. And then for majority of the talk, I'll focus on the NIMBLE study, how we are focused on qualifying biomarkers for NASH. And Detlef, thank you so much. You already shared some of the results, and I would not go over the results, but really talk about the strategies and how we approach doing these types of studies. So I don't think I need to talk to this audience about the public health significance of NASH, except to say that from a patient perspective, the diagnosis rate is poor. We really have no approved therapies, which is kind of terrifying because there's projected to be 3 million cases of NASH by 2030, and I think it's already the number one reason for liver transplant in women in the United States. So we are in a situation where unless we are able to easily diagnose and find treatments for the disease, we will be, we will have a significant public health burden more than what we have already. So that's sort of is something that's worrying all of us. But as we were focusing on what we need to do, it was evident to the group that had come together to discuss it that what we really needed was to identify the high-risk NASH population, those with more F2 of greater than or equal to 2, and maybe NAS greater than or equal to 4, and this is still about 10 million people affected, and these are the population most likely to progress to cirrhosis. So we would like to find a way to diagnose and enrich these populations with non-invasive biomarkers so we could enroll them in clinical trials and give them better care at care centers. And so that's sort of what we ended up as taking on as our charge, or our goal is how are we going to find this slice of the population without going into liver biopsies? Because as has been alluded to several times, it is a challenge to do liver biopsies for large scale applications, and it has been the biggest impediment for drug development. I think we all understand that it can be invasive and painful and carries a risk of small risk, but a risk nonetheless of mortality. We deal with sample variability depending on where the biopsies are being taken for. There's of course observer variability, and because it can only be done in special places, the capacity for who can do liver biopsies is limited, limiting our patients' access to liver biopsies and increasing the cost. So all of these reasons make it both a barrier for access to care and also for drug development, because we cannot do it frequently, and we have variability between sort of biopsies across the liver. So all of these reasons made it necessary to look at non-invasive biomarkers. And like I said, of course, we all agree we need non-invasive biomarkers, but the focus for NIMBLE has been to detect the at-risk NASH population, which are most likely to progress to cirrhosis, so that we are able to enrich a population that's going to be recruited in clinical trials. All right, so that's just to give you an outline of why we are working in this space. And so NIMBLE was born out of this need. And like most things that we do at the foundation, we wanted to be very clear about our goals, and we didn't want it to be to ask many questions, which are all possible, but focus ourselves on two goals that with the FDA biomarker qualification program, which you heard about earlier today, we should qualify non-invasive biomarkers that will enable two things. The first would be to diagnose and stage NASH, and this is specifically to identify at-risk NASH as well as patients with cirrhosis, so we can find that population that we can that are most amenable to intervention. And the second is to find biomarkers, response biomarkers, that will allow us to measure quantifiable change in NASH in a prospective study, so that we can read the response to any therapeutic interventions. So these were our two goals, and this is what this was the vision of the team. And I want to say that the team that put this together had leaders from academia, industry, biomarker developers, FDA, and we realized we needed a lot of different people to pull this together so as to meet our goal of qualifying these biomarkers. And that's why this program was brought in front of the Foundation for NIH and our Biomarkers Consortium. So in the next few slides, I will talk to you about the foundation, what our work is, and within the foundation, what the Biomarkers Consortium does. So the Foundation for the National Institutes of Health, or FNIH, is a nonprofit. We're not government. We are adjacent to NIH, and our goal is to create and lead alliances between public sector like NIH and FDA, and private sector, which would be large pharmaceutical companies, smaller biotechs, nonprofits, patient advocacy groups, etc. So we can build programs that can forward the mission of NIH to improve lives and find better treatments, while being translatable to drug development space. We were founded by an act of Congress in 1990. So this was our goal. Our whole purpose for our existence is to build these types of partnerships. And we've, I would say, been fairly successful in building our partnerships, because we do everything from bringing together partners to facilitating ideas, to developing them into full projects with milestones and deliverables, to executing those projects, doing the fundraising, and actually meeting the milestones. And the main thing that FNIH does in this whole process is we serve as the neutral arbiter, or sort of safe harbor, where we can hear from all of our stakeholders, whether they be from NIH or FDA, CMS, large and small pharmaceutical companies, and so people can work together as scientists to move things forward. And we work in a pre-competitive space to enable that to happen. These are some of our partnerships, and the Biomarkers Consortium, which I'll be talking about exclusively from now on, is one of those partnerships. And as you can see, it's not our largest partnership, but it does have a very specific goal of qualifying biomarkers. So the Biomarker Consortium works very closely with the FDA biomarker qualification program, in addition to other entities, to really bring established biomarkers towards qualification. And we have done several letters of intent and and in the process of doing qualification plans for biomarkers. One of the reasons the Biomarker Consortium has been, fairly successful for the last 15 years is we make our decisions in therapeutic area aligned steering committees. There are steering committees in 4 therapeutic areas. NIMBLE came from the Metabolic Disorder Steering Committee, and this is a subset of stakeholders that first ideate a project idea, and this is what is necessary, and this is a timeline in which this should be done, and then build a program design off of that. And NIMBLE was approved as a concept in 2018. It launched in 2019, and we are halfway through the project at this time. Across the Biomarkers Consortium and different therapeutic areas, we've had a significant amount of success. I think in 15 years, we have done 14 therapeutic advances. We have 9 clinical tools that are actively being used in drug development. We've contributed to 4 FDA guidance documents and qualified 1 clinical safety biomarker, in addition to publications, citations, etc. The consortium approach is also encouraged by the FDA to do biomarker qualification work. And this is one of the reasons why we have over 60 members in the Biomarkers Consortium, and they represent large and small companies, trade groups, as well as not-for-profit organizations. So like I said, NIMBLE came out of the FNIH Biomarkers Consortium and has grown into a really effective program. So in the next several slides, I will talk to you about the structure of NIMBLE, how we've envisioned it, and where we are in our process of qualifying non-invasive biomarkers. NIMBLE team is a really collaborative team of researchers. We have over 30 entities in NIMBLE. This is a true industry academic collaboration with very innovative team science. We have lots of meetings. We discuss all of our study design ideas, but the goal has always been to stay focused on what our primary deliverables are, which are to diagnose and stage biomarkers for diagnosis and staging of NASH, and biomarkers for that can show us response to interventions. And while sticking to those deliverables, we have built a large collaboration and are able to share some results most recently. And really the goal has been to advance the field of NASH forward so we can get to therapies quickly. And in order to do this, 2 things were very clear. The first is that we needed to have a very open collaboration across silos between the various sectors that are that have skin in the game. So we had pharmaceutical companies, biotechs, diagnostic companies, imaging companies, regulators, and academics, all of whom are involved actively and passionately care about this. But we wanted to make sure that we were in one room and talking to each other and using a common collected wisdom to meet our endpoints. And one of the things that we decided to do as in order to make that happen is that NIMBLE has all members of NIMBLE have agreed that all the data that's generated through the study will be put into public domain, either through manuscripts, publications, presentations, or through the FDA biomarker qualification program, and that no new commercial intellectual property will get generated as a result of this collaboration. And this has really helped our team to think outside the box and think creatively to implement the vision. So NIMBLE was established, like I said, to meet the high priority unmet need for non-invasive tests. And the 2 types of biomarkers that we are after. We wanted to make sure that in patients that have clinical risk factors for NASH, that we're able to diagnose NASH and determine the stage of the disease. and that in patients that are diagnosed with NASH, proven with biopsy, that we are able to detect and quantify the change of NASH status and fibrosis stage. And we decided to do this in a two-stage approach with a gating mechanism, which is common to all FNIH programs. So we are at the first milestone mark here. So we're just wrapping up our stage one. Stage one of NIMBLE has 3 studies. The first is a retrospective assessment of circulating or blood-based biomarkers, and the second and third are methodological studies that are going to look at ultrasound and MR-based biomarkers to look at repeatability and reproducibility across devices. And that's the stage one for NIMBLE. And our goal to meet our go-no-go milestone, which we are going to present next week to our executive committee. Our goal here is to make sure that we have robust and promising biomarkers that give us confidence that we can move into stage 2, which is a prospective study, which will include circulating functional and imaging biomarkers with bookend biopsies and some intervention that will allow us to see if those biomarkers are able to read the quantified change in NASH status. So this is where we are, and we are very excited about the next stage for NIMBLE. So it's slightly deeper dive into NIMBLE stage 1. NIMBLE stage 1 is evaluating both circulating and imaging biomarkers, like I said. The circulating biomarkers are going to look at sort of context of use is to look at diagnostic enrichment, so the patients who are at risk can be diagnosed and stage NASH so that they can be included into a clinical trial. And for the imaging biomarkers, we're going to look at systematic assessment of repeatability, reproducibility across across sort of manufacturers, Philips, GE, Siemens, and across field strengths to see that we know how we can measure, how we can equate the measurement read on one machine versus another, which of course is very important when you're doing clinical studies and you have multiple sites with different types of equipment. So like I said, our focused goal was to put in letters of intent with the FDA biomarker qualification program. And I'm happy to say that both circulating biomarkers and imaging biomarkers put in their letters of intent, which were accepted. The circulating biomarker letter of intent was accepted in February 2020, and the imaging biomarker letter of intent was accepted in April 2021. And you can look at both our submissions and the response from the FDA by going to the biomarker qualification page. So I'll focus on the circulating biomarkers because this is where most of the results have come in. We have wrapped up our study on the ultrasound biomarkers, and we are halfway, or we have started and towards the middle of our imaging MR biomarker study. So we should have results on those in about 6 months. But I'd focus on the circulating biomarkers and how we decided to study these panels, right? So we've heard about NIS4, OWL, ELF, PRO-C3, fibrometer, and so many others on various talks today. But we at the NIMBLE team decided to focus on these 5. And this was based on a cross-stakeholder assessment of which of the biomarkers that were available had the most significant analytical and clinical proof and validation that made the team feel like these had the best chance of succeeding. And so that's how we picked our biomarker panels for circulating and imaging biomarkers. And for for the at-risk NASH, we were looking at NIS4 as the primary panel, but for fibrosis stages 2, 3, 4, and cirrhosis, we evaluated ELF, ProC3, and fibrometer VCTE as well, and that's sort of and to see how because that was sort of the specific intended use population per the developer of those biomarkers. And in order to run the studies, NIMBLE collaborated with NIDDK's NASH Clinical Research Network. And I'll briefly go over the study design with you. You'll see the term CWS come up. This is a work stream within NIMBLE that is specifically working on the circulating biomarkers. For the study, NASH CRN contributed more than 7,000 samples from 1,000 unique patients. And each of these sort of sets of 1,000 tubes were sent to each one of the biomarker developers. And it's important to know that the samples were selected that were biopsy confirmed, and the biopsy had been done within a 90-day window. All the biopsies had been centrally read by the NASH CRN pathology committee using a pre-specified protocol, and so that information was all there. And then the relevant clinical data was shared by NASH CRN, and the biomarker data was shared by individual biomarker developers, all of who are blinded to each other, to a central third-party CRO who put it together and merged it and generated the results based on a pre-specified statistical plan that was developed by NIMBLE team before the data was sent over. So we wanted to be very, very clear in our handoffs and wanted to make sure that there was no possibility of contamination or influence of the results from the data that was going in. And this is sort of a more visual depiction of that. So if you look from the top left, the samples, like I said, over 7,000 samples across disease spectrum from F1 to F4 were acquired with the help of our colleagues at VCU and Arun Sanyal, who is the academic chair for NIMBLE. And these were split across the 5 biomarker developers. The data that was generated was shared with Cytel, who was our imaging CRO. There was metadata from NASH CRN per the SAP was shared once they had the primary data was with the statistical CRO, and that was together put into the first set of data. And we had, because of so many involved stakeholders, we had a priori set up our cascade of reviews. The NIMBLE and CRN team reviewed it first, followed by the whole NIMBLE project team, which had all the sponsors and FDA, followed by the collaborators or our biomarker vendors and biomarker developers, and then that was all put together. And once we were all in agreement, we are at step 8 right now, which is the data is being presented and the manuscripts are in development, and we are hoping in early next year we'll be able to have discussions about the qualification plan with the FDA. So these are the results. I think you've seen this one table on the left before, earlier in this set of talks, but you can also find this on the FNIH website. This was presented by Arun at the ASLD meeting, and really shows the results for both NASH diagnosis as well as fibrosis stages for each one of the biomarkers. So I'll just briefly conclude for you the results. You please feel free to review them at leisure. NIS4 met the a priori criteria of diagnosing NASH, diagnosing NAS greater than or equal to 4, and fibrosis stage of 2 or higher. So in sum total, NIS4 was effective in diagnosing at-risk NASH population. OWL test was able to identify at-risk NASH with sensitivity of 63 and specificity of 75. We are working on doing more evaluations with the OWL panel. The ELF and the fibrometer VCTE test met the criteria for detecting clinically significant fibrosis, advanced fibrosis, and cirrhosis. And as Detlef mentioned, the ability to predict the fibrosis stage improved with the stage of fibrosis. So we got better results the longer the more complex the fibrosis stage was. PRO-C3 was superior to the unit line, but was not significantly superior to FIB4. It performed well, just not significantly superior to FIB4. And FIB4 actually had a fairly robust diagnostic characteristic for all the fibrosis-related endpoints. So this is just a summary of the results. I encourage you to review Arun's slides from ASLD, read up on it, and we are in the process of writing our manuscript. Hopefully, we'll be able to share more soon. So I just wanted to share a conceptual slide for why we feel that this type of work represents really sort of the best type of team science. And this is just because we've been doing this for a long time at the foundation. You know, stakeholders have different needs and interests. When the stakeholders are academic, their interest is to publish, find its research funding, expand expertise and possibilities. And if the stakeholders are from industry, they're interested in creating a product, share risk, reduce costs, you know, expand expertise, applications. And the public sector stakeholders, FDA, NIH, as well as the patient-engaged voices are interested in communicating needs, building consensus, and looking forward to the future. And so a project is a public-private partnership is successful only if it fits within the small little triangle of the overlapping Venn diagram. And so at NIMBLE, we support our academic scientists with their publications. We are expanding access to data and the research that we are doing. We are helping our industry partners de-risk their decision making based on the results that are coming in. And we are working closely with FDA, NIH, and patient-centric organizations to really start talking about how this can be really meaningful in patient care and drug development. And this last slide, how do we manage such a web? We have a very stringent conflict of interest policy, and we do joint decision-making as long as everybody is non-conflicted. So we have a very well-defined conflict of interest policy, and whenever there's a decision to be made, everybody who's a part of NIMBLE makes a decision as long as you're not conflicted. We developed our SAP a priori, and all data and analysis were handled by our CRO. So we were all very, very clear that we were doing the best we can to get the cleanest results. And this is all supported by a fairly complex web of contracts and negotiations that the FNIH manages with all the biomarker developers, the academic institutions, various CROs, and NASH CRN. And it is really a true public-private partnership. I just wanted to thank the overall leadership, the imaging biomarkers leadership, the circulating biomarkers leadership of NIMBLE. And this is a picture from our launch from 2019. We've gotten actually bigger. We have some more partners and collaborators since then, and they're all represented here. These are all of our NIMBLE members. Either they are sponsoring partners, or they are, you know, on the steering committee as you know, giving us regulatory insight, or they are device companies or imaging companies, and we all sort of work together to hopefully come up with a solution. And with that, I will take any questions.
Thank you very much for your presentation, Tania. I think everybody will agree with me when I say it's a really great presentation to end the summit. I do have a couple of questions to ask you, if that's all right. First of all, just wanted to ask you, how can one participate in NIMBLE?
Yes, so we have just, like I said, we just finished our stage 1, and we are going to stage 2, which is going to be a prospective study. We have 14 sponsoring partners on NIMBLE right now. We are welcoming more, but it will depend on the study design and structure and what is necessary for that. For people who have biomarkers they would like to get tested, or technologies, digital measurements, we are encouraging those applications. It will all be a part of stage 2. So you'll have to sort of just, wait with us a little bit because of the joint decision making. Things take a little bit longer, but we are hoping in by the end of Q1 2022 we'll be able to do that. So if you're interested, you should just reach out to me and I'll be able to direct.
Lovely, thank you for that, Tania. I also wanted to ask, where are you with the biomarker qualification efforts?
Very good. So, we put in 2 LOIs, one for imaging biomarkers and one for circulating biomarkers, both for the same context of use. Our goal was always to just combine and see what a combinatorial approach would be best. We are in discussions with our FDA colleagues, and it might be that we will go forward with submitting individual qualification plans for the circulating and imaging biomarkers. We are also looking at combining or looking across all the circulating biomarkers and seeing if it improves the reads, and the same for imaging. So that is also something that is being planned for early next year, but we'll be able to, we'll put something out in media when that happens.
Lovely, thank you for that, Tania. And just my final question is, how did you select the biomarkers for the study?
Right, I think I may have addressed that already, but that was a difficult one because we had there were so many promising biomarkers, and we had to come up with a set criteria that the core team initially agreed that these were the top 5, but we also realized that science changes and we want to stay current, and so we've got additional biomarkers that people have reached out to us and they want to be a part of NIMBLE that are being evaluated as exploratory biomarkers, and I think there's going to be a time to do that as well. We're just focused on getting our qualification plan in and getting our stage 2 started, but I think we would like to keep up with what is happening and make sure that we are being relevant to what is happening in the field.
Lovely. Thank you so much for your participation, Tania.
Thank you.